Hello, everyone. Welcome to Chem 1115, Chemistry and Society. Today, we are talking about Chapter 13 of our textbook, and these are the notes that go along with Chapter 13. In Chapter 13, we're learning about genes in life, as your textbook calls it. What is genetic engineering? What challenges are genetic modification solving? What role does DNA play in life? All of that good stuff. So here is your reflection for this chapter that you will be doing on your own. And let's talk about some definitions. The first one is genome, and then within that is a gene. So the entire DNA sequence of an organism is called its genome. So if you took every single piece of your DNA and you sequenced it, that means you got the order of the bases for all of you. That would be your genome. But within your genome, you have short segments of DNA that code for specific proteins, or as we would say, specific traits, then that's called a gene. And then if you alter a gene, then you alter those traits. And inheritable just means that it can be passed from one generation to the next. So here's a figure that is an animated genome. I encourage you to go check out. So these are probably review words that you've heard from your biology courses in the past. So I'm not going to spend a ton of time going over them because I'm guessing you've heard these words before. All genetic information is stored in the nuclei of all your cells. So your cells all have the same DNA. What differentiates your cells from each other is which traits are expressed. So if you are a liver cell versus a heart cell, right, what makes the liver cell different from the heart cell? Well, they have different properties, obviously, but they actually have the same DNA. It's just certain parts of the DNA are silenced and certain parts of the DNA are turned on, so to say. And proteins control that primarily. So each nucleus in a human, I should say, within humans, because we're not talking about corn. Corn has different numbers of chromosomes. Each nucleus in the human body contains 46 chromosomes. So again, different organisms have different numbers of chromosomes. And so what is a chromosome? It is an extremely compacted version of DNA. If you took your DNA molecule and you stretched it out, it'd be about 2 meters tall. To give you a frame of reference, a meter is approximately the distance from the floor to like your belly button, if you're of average height. So just imagine it's floor to ceiling, one DNA molecule. It's a big, long molecule, okay? It's a polymer. We'll get to that in a minute. But to fit that giant molecule inside the nucleus of all of your cells, it has to be what's called supercoiled. So it folds up on top of itself very neatly into chromosomes. And again, if you go on to take biochemistry or molecular biology, you'll study that detailed process, very, very, very detailed process. But here we're just talking about chromosomes as being a compact version of DNA. And DNA stands for deoxyribonucleic acid. Again, if you go on to take biochem, you'll learn what that means, and you'll actually see the molecule and why it's called deoxy. So if you had all your DNA spread out, it would be gigantic. So it has to fit inside the cell, and that's where chromosomes come in. So this is important for you to understand because we've talked about polymers a lot this semester. DNA is also a polymer. It's made out of nucleic acids. So what is the monomer of DNA? It's nucleic acids. And there are 4 monomers in DNA: adenine, which we represent with a capital A; guanine, which we represent with the uppercase G; cytosine, which we represent with the uppercase C; and thymine, which we represent with the uppercase T. So DNA is a very long polymer. And what is the polymer made of? These 4 monomers. And here are their structures. I'm not going to make you memorize that, but future classes, yes, you will. So here's a picture showing DNA as a polymer. These just link to each other. It's called a sugar phosphate backbone. You don't need to know that for a test, but you will in future classes because the bonding is occurring between this phosphate molecule and this ribose sugar. And how you can tell it's DNA is by looking at that carbon right there, by the way. If this were RNA, that would be an OH. Just fun fact. So DNA is double-stranded. So that means you have two strands of DNA. There's strand number 1 and there's strand number 2. And DNA is called complementary. So that means anytime you have a C on one side, you have a G on the other. And anytime you have a T on one side, you have an A on the other. So A and T complement each other and G and C complement each other, which I'm guessing you remember this from biology as well. So they are your complementary base pairs, A and T, G and C. And one of the things that I want you to understand is that these molecules, the C and G, for instance, aren't bonded to each other. Those are not chemical bonds. These are called intermolecular forces. They're strong forces of attraction, but these are not actual chemical bonds. All of these that you see right here represented with the black line, those are chemical bonds, whereas here these are just attractions. And that's something to keep in mind for future classes. I won't ask you that on a test, but just fun facts. The two strands of DNA are not actually chemically bonded to each other. That's why you've got these dashed lines. Even though some textbooks represent it as like stairs in a ladder, they're not actually bonded to each other. Just fun fact. Okay, so what is genetic engineering? It's the direct manipulation of the DNA of an organism. And so the simpler the organism, the easier it is to manipulate, right? So unicellular bacteria are the easiest organisms to manipulate. And so what you do with bacteria, they have very simple DNA. It just floats around in these little circles, and you can easily manipulate them. And again, if you take molecular biology or biochemistry, you'll learn some details about this. I remember when I took genetics, we were learning how to genetically modify bacteria to make them glow in the dark, fun stuff like that. So you can genetically modify the simplest organisms, pretty straightforward. And so you hear a lot about this in the media. Why would you do genetic engineering? And here's some of the reasons: to make crops more resistant to disease or stress, higher yield, making more enzyme, or using an enzyme to generate a drug. That's actually what was behind the development of insulin as a commercial product. Developing vaccines out of edible products even, right? So there are lots of reasons why genetic engineering is used in our society right now. And we could spend all day talking about just the reasons behind genetic engineering. So GMO stands for genetically modified organism. And so there's a lot of controversy within social media and in the news and in public opinion about GMOs. So one of the arguments that you'll see, it's not natural, it causes allergies, it can cause cancer, it can cause overuse of pesticides and herbicides. And so here are some counterpoints to think about. It creates superbugs, superweeds, they can't replant them. And again, here are some counterpoints for you to read over. My job as a teacher here is to enable you to make your own scientifically based decisions. Okay, I'm not here to tell you my opinions on these things. I'm here to teach you how to form your own opinions on these things. And as a scientist, you need to be able to read the data, interpret the data, and draw your own conclusions about the scientific information. So for you, you need to read these counterpoints, think about them, and really digest them. Think about them as a scientist. Think about them in terms of all of the things we've learned this semester, because this course is called Chemistry and Society, and this is one of the biggest hot topics for our society right now. So for you, as a scientifically literate person, you need to think about all the chemistry that you've learned this semester and then apply that to these hot topic situations. So again, read the data, interpret the data, and then draw some scientifically based conclusions. I hope that now that you've completed this course, you are able to think like a chemist. And I hope that I've sparked your interest in wanting to take future chemistry classes or future science courses, or maybe you've even decided to change your major and become some version of a science major, which is awesome if you do, but you know what? If you still don't like chemistry, that's okay too. I understand. So that's where we're going to end chapter 13. If you have any questions, please feel free to reach out to me, and I hope you have a good day. Bye.